The last section of this webinar is about Listeria control verification. So in terms of requirements, as part of FSMA preventive controls, food facilities need a monitoring program. So if contamination of a ready-to-eat food with Listeria, for example, is a hazard, then you need to do testing and environmental samples. And this applies to you if you're considered a food facility, and you're considered a food facility if you're required to register with Section 415 of the FD&C under preventive controls of FSMA as well. And if you fall under the PMO, you can actually use that. I'll talk about that in a second. So your main sort of resource for Listeria control should be the Listeria draft guidance. It is currently a draft, but a lot of agencies are sort of using this as, you know, the standard for right now as they're getting it finalized. So you should definitely be reading this for all of your Listeria questions in terms of how to regulate Listeria. So it's only 85 pages. I promise it's a quick read. So definitely check that out. I want to highlight a couple of things that's in there. One of the things that they have is cleaning and sanitation. So when you're reviewing cleaning and sanitation SOPs, it should really have a lot of these steps. And in particular, it should really focus on the cleaning part. A lot of people like to just take their drain and dump sanitizer down that, and that's not going to clean the drain. It's actually 90% cleaning, 10% sanitizing. So you should really be using detergents and scrubbing to get surfaces clean. And then the very last step should be sanitizing. Another thing that I want to note that's in this draft guidance is they talk about keeping dry rooms as dry as possible, because water is really critical for the growth of Listeria. So if you've got a dry room, you don't want to be getting it wet, and definitely not during production. So you can really do a dry clean in those drier rooms. At minimum, for verification, you need written records. For example, you need to measure and record sanitizer concentration, and you should be doing visual inspections as well after cleaning and sanitation. It's also recommended to do ATP testing on food contact surfaces and total plate counts as a verification for cleaning and sanitation. So the draft guidance talks about the goals of environmental monitoring, and so the goals are to verify that your control programs are effective, to find Listeria and harborage sites if present, and to ensure that corrective actions have eliminated Listeria and harborage sites when in your plant. So talking about that persistent, resident, pet Listeria, all of those is referring to this third bullet. There's also a section on what your written procedures should be. So for example, they should be scientifically valid. They should specify species versus monocytogenes, identify locations, identify timing, identify the tests and the labs, and corrective actions. And for those of you who are using the PMO, which I'll mention in a second, these are actually the same. So again, this is another example of a slide that could really be its own hour-long webinar, but here's really what you need to do if you're starting a pathogen environmental monitoring program. It should really involve at least, at a minimum, all of these steps. And again, you could refer back to this in the recording. Another thing that comes up in the draft guidance is that 5 sites for sampling should be food contact surfaces, and 5 sites should be non-food contact surfaces. So I caution you to be careful with this one, because if you do end up doing 5 sites for food contact surface, I want to make sure that you're aware of the things that you need to do in terms of corrective actions for a positive. You have to be aware of the consequences of a positive. Don't just test the sites and then ignore it if it's a positive. So if you have a food contact surface positive, you have to have a plan in place of what you're going to do. You obviously need to retest, possibly hold or recall product until you find out what's going on. So here's a little flow chart that's actually present in the draft guidance. For low-risk food, when I visit food facilities that are really good on food safety, If they're a low-risk food, they actually don't do a whole lot of food contact surfaces, and they do much more zones 2 and 3, so the surfaces next to the food contact surfaces. But if you're a higher risk food, for example, soft cheeses, for example, you should do food contact surface testing, and it should be negative. But again, have a plan in place so that you're not just testing and hoping that it's negative every time. I keep mentioning the PMO. If you're covered under the PMO, then you should be covered in terms of Listeria. You just need to check page 396, Appendix T, preventive controls for what you're supposed to do with regards to Listeria environmental monitoring. Finally, I want to talk to you about how you might check to see if your sampling plan is working. So here's an example of research from 7 different cheese plants where we did routine sampling. And so you can see the prevalence from several months of sampling. And so if I were to just look at this based on the prevalence, I think I would want to eat at facility F or facility I, because, or even facility J, because they look like they've got the lowest prevalence. And I think I would be most worried about eating cheese from facility H, G, and E, for example. But when we sent in, so all of this testing was done by the food facility themselves. And so when we sent in outside experts to pick sampling sites and to actually do the sampling, sites from zones 2 and 4 over 6 months, we actually found out that the prevalence was quite different from what they were finding in the routine. So facility F, out of 334 samples, they didn't find anything when they were doing the testing. But when our outside experts came in, they found 6% prevalence. Same with J, there was also a huge jump. So this is a great way to validate whether or not your routine sampling is looking in the right places and is actually doing the swabbing correctly. So you want to be a facility that's green, where someone from the outside actually has a harder time finding Listeria, because really the people who are doing the sampling in the facility itself that are familiar with the facility should be better at detecting it and finding it than someone coming in from the outside that's never seen the facility before. So to summarize, Listeria can contaminate ready-to-eat foods, causing illness and death. Verification and validation of cleaning and sanitation programs is essential, as Listeria can survive in a food facility over time in those niches where cleaning and sanitizing is ineffective. And lastly, most ready-to-eat food facilities are required to have a scientifically valid sampling program. So that's all I've got, so maybe we can open it up for some questions. Let's see what we've got.
Hey, Genevieve.
Yes.
Hi, this is Renato. We have questions in the Q&A,
Okay.
and we also have a question in the chat. Do you want me to read, or you want to?
Yes, that'd be great.
Okay, so in the Q&A, we have a comment from Bill. He says, the sampling sponges shown are fibrous cellular material, and the broth mentioned is DE broth. And then he has a question. What are your thoughts about high-capacity neutralizing broth and polyurethane sponges versus the above mentioned?
So I'm less familiar with the other type of neutralizing broth and polyurethane sponges. The ones that I mentioned are the ones that we typically use. They seem to work really well for us. So if you're considering something else, either because you think it works better, or because it's just a better price, or because your lab is better equipped to handle it, one of the things that you can try is using both in food facility and seeing which seems to detect Listeria better, and then just choosing whichever one works better for you. But as long as it has that neutralizing broth to neutralize sanitizers, and it has the surface area that's going to help you pick up Listeria if present, then you should be okay.
Great. So the other question was in the chat. It's from Alicia Johnson. She says, I was told that Listeria monocytogenes cannot grow in food with a pH of 5.0 at 41 Fahrenheit. Is that true? I thought, yeah, I thought it was able to grow down to a pH of 4.4 at a temperature of 41 Fahrenheit.
Yeah, so it can. So if you check, I think it's the FDA. I think those are the right numbers, 4.4 and 41. I would have to check to be sure. I don't have them in front of me. But yeah, Listeria can grow in some pretty extreme conditions. And that's not to say it grows well, for example, at pH 4.4. That's really the limit. But it can grow, and it can definitely survive. So you can use acid as a way to control, but it has to be pretty extreme. Hopefully that answered that question.
We have another question that pop up right now. Are you aware of Listeria associated with cut vegetables?
So I am aware of Listeria in facilities with cut vegetables. I'm trying to think. There's been several produce outbreaks, and I'm trying to think of one that's cut vegetables. I'm guessing you mean besides lettuce. I guess lettuce would be a good example of Listeria with cut vegetables. Um, yeah, does that answer that question, Listeria? Yeah, oh, besides lettuce, okay. Yeah, I can't think of any off the top of my head of cut vegetables, but it's definitely in those operations. I do know that 100%. And just because there hasn't been an outbreak detected yet doesn't mean there hasn't been an outbreak with cut vegetables and Listeria.
Okay, yeah, there is a comment now. There have been Listeria monocytogenes in frozen vegetables.
Right, yes, frozen vegetables. And that's definitely an issue when we're talking about smoothies, for example. So a lot of cut vegetables are being used in ways that people didn't originally think that they were going to be used. And smoothies is a great example of that. People love throwing chopped vegetables in their smoothies in the morning, and then they have them sitting in their car on their desk all day, and Listeria can grow and definitely become a problem. So yes, thank you to whoever sent that in.
Right, that was then, and she also asked, what about presence of multiple species of Listeria in a firm? How is that associated with the presence of Listeria monocytogenes in a firm?
Yes, so we sort of saw this with that deli example where we had the blue type of Listeria, and so that was the one that was the major issue because we kept finding it. But there's definitely a chance you're going to find not only multiple types of Listeria species, but also multiple types of Listeria monocytogenes. So that's definitely something to be aware of. Even within one sampling site and one sponge, you might find multiple types of Listeria. So that's definitely an issue as well. So what we found is that you might even, if you're looking for an outbreak strain and you're not finding it, you may even need to select multiple colonies for whole genome sequencing in order to find the one that you're looking for. Just because multiple types of Listeria can live together. In terms of your response to those, if a food facility has multiple types of Listeria, you should be more worried about the overall prevalence. Are you finding it in a lot of different spots? Are you finding it in high risk areas, for example, food contact surfaces? And if so, that's when maybe an outside cleaning and sanitation team should be brought in to do a deep clean of the place, and maybe cleaning and sanitation SOPs should be reviewed. But if you're finding like one or maybe even two Listeria in a food facility, that's not uncommon. So that shouldn't be, um, reason to come down hard on that facility, for example.
So following up on that, Genevieve, another question, what's your advice, well, not a question, what's your advice for a facility that has found Listeria over a longer period of time?
Right. So It again kind of goes back to the prevalence and where they're finding it. So if they're continually finding it in, for example, a sandwich juncture on a piece of equipment, and they keep finding Listeria, even if they're not sequencing it, they don't know what type of Listeria it is. That's a case where they either need to review cleaning and sanitation, where they need to disassemble that during cleaning and sanitation, or they need to get rid of that piece of equipment. Obviously, that's not a perfect solution, but if it's a place where Listeria is continually able to survive and be detected, then that's an issue, and they need to address that, no matter what the sequencing type of Listeria. If you're in a facility where you're finding different types of Listeria every single time you go, and it's more than one or two, then cleaning and sanitation procedures again need to be reviewed. So yes, I guess it's on a case-to-case basis, but if they're continually having Listeria found in a food facility and high numbers of it, then yes, you need to review that those procedures. Hopefully that addressed that. Feel free to send a follow-up question if I didn't quite answer your question.
I think it did. We don't have any other question right now, but I would like to remind everyone that we have a survey. I posted a link, and I'm actually going to post right now again in the chat box. So if you can go to the link and complete the survey, that would be great. Let me see if there is any other question. Okay, it looks like we have. How can you verify supply if it is associated with a product that is brought into the facility?
Okay, so that's a good question. So we have seen cases where you'll find Listeria from two different facilities that actually appear related, and so oftentimes that's caused because they have a common supplier. So what you need to do is you need to go a step further and figure out where that supply is coming from. Is it being contaminated on a common truck? Is it coming from the same field? If it's produce, is it coming from a previous storage facility, or otherwise just a previous facility where a food is coming from? And so you sort of had to follow the supply chain to the previous location and do sampling there and sort of investigate and do an inspection or investigation. So that's actually a great question, because you really do have to keep that in mind. You can't just assume that because you find the matching Listeria to a case that it's because it's from that facility itself. It actually could be coming in on product from another location. Hopefully that answered that question. But in order, the facility, you should be able to ask them, where is this coming from? If they can't answer that, then that's a problem.
Thanks. Let's see. I think there is another message. Okay, so I think we don't have any more questions then.
Okay.
Okay, thank you. Thanks, Renato. Thank you so much, Genevieve. That was really excellent, and I really appreciate your time and your expertise and for being willing to collaborate with us on this project. Again, I wanted to recommend that people fill out the survey, because that'll help us to understand, whether or not you found this useful, and if you have any comments about how to make it better, we would appreciate that as well. Additionally, if I did send you a sign-in sheet to have you sign in, if you were hosting the webinar, with a large group, it'd be nice for us to know exactly who in your group attended, and we'd also like to have a list of the attendees so that we can send out any further information that we may have. So anyway, I'll end the meeting now, and thank you again, Genevieve, and thank you, Cornell, and thank you, Renato.